The 2009 Albert Lasker Basic Medical Research Award honors John Gurdon and Shinya Yamanaka for their breakthrough discoveries concerning nuclear reprogramming, the process that instructs adult cells to form stem cells.
One can look around among a whole range of cell sizes and choose one that's just the right size for the pipette that you have.
In 1956, John Gurdon, a graduate student at Oxford, intrigued by the research of Briggs and King, who transplanted a living nucleus, decides to replicate their experiment.
There's no question they deserve great credit for having discovered how to transplant a living nucleus out of one cell into an egg and get development from it. And this had never been achieved before.
From their experiments, Briggs and King concluded that permanent nuclear changes occur as cells specialize.
What Briggs and King said is that as development proceeds, the nuclei of cells seem to lose their ability to participate in normal development. They can no longer make normal tadpoles from injected eggs.
Dr. Gurdon decides to further investigate this finding. However, the design of his experimental protocol has several important differences, including the innovative use of a genetic marker.
And the overall conclusion was coming out in a different direction, namely that as cells differentiate, nevertheless, their nuclei remain what we call totipotent, can still reprogram an egg.
Micropipette tip can be passed into the middle of the egg.
Gurdon had not set out to contradict the findings of Briggs and King, but his findings propose a very different reality from the mainstream research of the time.
The old idea, which was still favored by some people, including actually my own supervisor, was that as cells differentiate, they lose the function of genes no longer needed. So as this embryo forms skin, it would lose the function of brain genes or heart genes.
As Gurdon's results were at odds with the findings of the two esteemed developmental biologists, the scientific community doubted his results.
I was essentially a starting graduate student who was, as you put it, challenging the results of highly respected, very innovative workers. And why should you believe a graduate student when these eminent and distinguished, very successful people had already done the experiments and reached a different conclusion? So there was a considerable skepticism and disbelief.
Dr. Gurdon's research findings show that the genome remains intact, which he further demonstrates by growing fully functional tadpoles from transplanted nuclei of adult cells.
Did we understand the ramification? In one sense, yes, in another, no. In the sense, yes, if you were persuaded, as I felt I was, that the fundamental principle is there, that the genome, the set of genes is conserved through the whole of cell differentiation. that to me was a very important conclusion, because it tells one how you try to understand how development occurs. It's nothing to do with losing genes or permanently switching them off. It's a question of regulating what we call transcriptional activity of the genes. Turn them on and off, but they're always there.
Dr. Gurdon's findings now start a new era, with many scientists building on his discovery of gene conservation. But it is not until 5 decades later that his work leads to Dr. Yamanaka's breakthrough. In 1999, Dr. Shinya Yamanaka, intrigued by Dr. Gurdon's research on gene conservation, reasons if all the genes are still present, it must be possible to instruct adult cells to form stem cells.
At that time, I was like 35 years old, so I thought, well, I could enjoy the rest of my life with this project. So it was very, very surprising even to us when we were able to make this within 5 years.
Dr. Yamanaka begins the search to identify all the factors that would be important for the maintenance of pluripotency in mouse embryonic stem cells.
We added all the 24 candidates into one petri dish. And after a few weeks, we saw like 10 colonies, which looked like ES cells. So that was the moment, we thought, wow.
For Dr. Yamanaka, it was a moment of hope, but as a scientist, a moment of incredulity.
We were just surprised. But at the same time, we thought it must have been some kind of mistake. So I asked him to repeat experiments again and again, but it always worked. And I asked another postdoc to do a very similar experiment, and it worked. So we thought, wow.
To determine how many of these 24 factors are actually needed, Dr. Yamanaka's graduate student devises an experiment adding all 24 factors and then eliminating one factor at a time.
So he had 24 combinations. And by that simple experiment, it took us only like a few weeks before we can actually narrow down into 4 factors.
This extraordinary breakthrough creates new frontiers for research. The induced cells resemble embryonic stem cells in their appearance and growth characteristics, both in test tubes and animals.
So we named these new stem cells iPS cells, induced pluripotent stem cells. So we and others have been comparing ES cells and iPS cells. And we found they are very, very similar, almost identical.
Dr. Yamanaka's discovery provides an amazingly simple technique to do something which was once thought to be impossible.
It's so easy, simple. All we need is 4 or 3 factors.
It is a rather amazing phenomenon, and I think that does eventually have considerable repercussions for experimental and therapeutic possibilities.
It's a science, so it's almost impossible to predict. You know, I expect some diseases like heart disease, heart failure, and some retinal diseases, maybe a good candidate. However, again, it's almost impossible to predict.
Dr. Gurdon and Dr. Yamanaka's accomplishments create a new world of nuclear reprogramming, fostering innovative strategies for combating disease, understanding the normal developmental process, and for pursuing embryonic stem cell research.